Dear students, Module 25. Oncogenetics. Properties of malignant cells and the types of genes, the proto-oncogenes and oncogenes. The main objectives are to know about the malignant cells and genetics of cancer, to understand the role of oncogenes, to explain the activation of proto-oncogenes to oncogenes, to know about the progression of cancer, to clarify about the oncogenes and cancer. Introduction. Our body functions associating with many organ systems. Organ systems consist of specialized tissues. There should be a proper amount of cells, and any extra cell can be deleted by the regulatory process called programmed cell death, which leads to the apoptosis. The growth and the death of the cells mainly depend on the genetic machinery. Any dysregulations that occur in any genetic control may lead to the death of the cell or the overgrowth of the cell. Some chromosomal instability and many mutations can result in the formation of the cancerous cells. The single mutation in the cell generally will not lead to any cancer. Mutations are common in our body, which brings the variations. When the specific mutations occur more times in a single cell, can develop the malignant cell. There are some external factors which can induce cancer known as carcinogens. The environmental factors like chemical carcinogens, food habits and radiation can affect in most cases of cancer. In most of the tumor, the carcinogens induce cancer by damaging the DNA molecule. The damaged DNA and the resulted mutations will accumulate in the cell to mislead the pathway of cell divisions. In the case of disease, there is a continuous growth of the cells. There are some essential genes in cancer genetics which are responsible for the progression of cancer. Mainly, there are two types of genes involved depending on the mutation that occur in those genes. It includes the oncogenes and the tumor suppressor genes. Hence, this episode explains further the following subheadings: A. Properties of malignant cells. B. Proto-oncogenes. C. Oncogenes. D. Proto-oncogenes to oncogene cancer progression. E. Oncogenes and the cancer. Property of malignant cells. It is well established that cancer has a genetic basis. The tumor cells, when cultured in the lab condition, they start dividing these daughter cells have cancerous property. Hence, it was confirmed that genetic factors which get transmitted to the daughter cells during the cell division. At the meantime, it was found that some of the biological agents like viruses induce cancer by the formation of tumors. The proteins produced by the viral genes have a role in the formation of cancer. Some of the carcinogens which are responsible for the formation of cancerous state can induce the mutation in the normal cell genome and cause the tumors. All these confirmations are done experimentally to ensure the genetic basis of cancer. It was found that certain forms of cancer could see running through generation in some families. Some of the rare types of cancers having the dominant condition could be inheritable. All kinds of cancers have genetic defects. Some people can get during their lifetime by some of the acquired mutations or some chromosomal abnormalities. Some cancers in the white blood cells, leukemias and lymphomas include the chromosomal defects. Cancer development occurs when mutations occur in critical genes. This will lead to the unregulated division of the cells. Cancer can be benign or malignant. If the tumor is formed and the cells from the tumor move towards the surrounding tissues to form the multiple tumors again, then it is termed as malignant. If the cancer cells from tumor stay in that region without invading towards the surrounding tissue, are termed as benign. Malignant, usually dangerous, and tumors can spread in different parts of the body. The process of metastasis occurs where the tumor cells will move into different regions of the body and the new growths of tumor cell arise. The continuous specific mutations will form malignant cells in a single cell. There should be 10 to the power of 7 of mutation rate for a gene per cell proliferation. Cancer can be progressive by the following two mechanisms. One, mutations that occur in the cell will lead to the active proliferation of the cells to form the mass of cells. These formed populations of cells subjected to the next mutation. 2. Other mutations will alter the genome stability. There can be a change in DNA stability or chromosome stability. This will increase the entire mutation rate and lead to the progression of cancer. Initially, there will be an accumulation of the affected cells or the benign tumor formations. The malignant tumor cells show the chromosomal abnormalities when examined under karyotyping. Some of the properties of the malignant cells are they can increase in size more rapidly, they lack the capacity of differentiation, they can possess abnormal membrane and other cytoskeletal proteins. They are insensitive to anti-growth signals. They can invade the surrounding tissues and metastasis. Proto-oncogenes. Proto-oncogenes are homologous to the viral oncogenes. It was found that viral oncogenes are evolved from the normal cellular genes. The mutation occurs in the proto-oncogenes, result in the formation of the oncogene, which are the more active form. The normal function of the proto-oncogene is to encode proteins involved in the cell divisions and growth. It is more critical for the normal function of the tissues, organs, and the development of the body. Proto-oncogenes play an essential role during the early stages of development. The cell divisions and differentiation occurs during the embryogenesis. These genes participate actively in the formation of the proteins required for normal development. Once the development process is done, these proto-oncogenes get turned off. Some of the proto-oncogenes can be growth factors, growth hormone receptors like epidermal growth factor receptors, EGFR. Some can be transcription factors, MYC, helps in the cell functioning pathway. C. Oncogenes. The genes responsible for the normal proliferation of the cells are called proto-oncogenes. When the dominant mutation occur, which are the gain of function mutations, will lead to the more active form of the gene. Mutation make the oncogenes to the more active form. A single affected allele is enough to change the phenotype of the cell. These oncogenes are initially found in the retroviruses. Some of the viruses have the complex genome, while other viruses like retroviruses have simple genome composition. Usually, the genome of the retrovirus has three following coding regions: GAG, POL, and the NV units. GAG encodes for the core proteins, POL encodes for the enzyme called polymerase, and N encodes for the envelope proteins. These coding units are critical for the retroviruses to infect. It was found that the acute transforming capacity of the retroviruses is due to the presence of one more extra gene called an oncogene. The oncogenes are copies of the mutated proto-oncogenes. Some viruses can induce cancer, but not all the tumors are depending on the viruses until the activation of their proto-oncogenes. If the fibroblast cells from the mouse are treated with the DNA fragments of the tumor from the human, some of the cells will be transformed. These cells are further analyzed to detect the oncogenes in the transformed cells. It was found that the viral oncogenes are derived from the cellular platelet derived growth factors B. The growth factors usually help in the proliferation of the cells. There are group of oncogenes derived from the viruses encodes the protein which are act as the transcription factors. Some of them are VJUN, VFOS, VMYC genes. The proteins formed are homologous to the cellular proteins. They can bind the cellular DNA sequence and regulate the process of transcription. Functions of the oncogene show that they have control over some of the DNA binding proteins, secretory growth factors and cell cycle components. Hence, these oncogenes can take control over the other cellular genes and regulate the cell functions. The product of these oncogenes can act as the binding proteins and some as signal molecules to regulate the functions including cell divisions. Proto-oncogenes to oncogenes cancer progression. The activation of the proto-oncogenes leads to cancer progressions. Activation of the proto-oncogenes includes the following. A. Activation by amplification. There will be the presence of many copies of oncogenes in case of cancer. These hundreds of copies will found as a small chromatin bodies. These structure found as small chromosome fragments without the centromere. They will make continuous gene replications and the formation of more gene products by the high level of gene expression. This will help in cell growth. The copies of the genes can also get inserted into the normal chromosome. These are termed as homogeneously stained regions, HSRs. These regions on the chromosomes will not show any dark or light bands. Examples: amplification of proto-oncogene HER2, ERBB2 and MYC in case of breast cancer, NMYC in case of neuroblastomas and ERBB2 in case of breast and ovarian cancer. The comparative genomic hybridization can detect the amplification of the oncogenes leading to cancer. The DNA mixtures of normal and the tumor cells are subjected to the hybridization. Both the DNA fragments are fluorescence labeled. Then the hybridization shows the signals by the fluorescence colors. B. Activation by mutation. The mutations like substitutions, insertions and deletions will alter the protein structure. These are the most common point mutation to occur where the single amino acid changes in the polypeptide chain. These variations will form the activation and the number of production of mutated proteins required for cell proliferation. A mutation leads to change in the regulatory regions of the protein. For examples, mutations occur in the gene ERBB, ROS, TRK oncogenes leads to the progression to the tumor formation. Most standard point mutations occur in the ROS family of proto-oncogenes. ROS proteins involved in the signal transductions which are essential for the controlled regulation in the cell differentiation, amplification and cell migrations. The G protein coupled receptors help in the signal transduction. When the ligand binds to the receptor, it will further promote the binding of GTP to the ROS protein. The GTP ROS will send the signal. ROS protein family have the GTPase activity. Hence, the signal will turn off soon by converting GTP ROS to GDP ROS. When these proto-oncogenes get mutated by the point mutation, the alteration in the formation of protein products lead to the more activation of the mutated proteins. This will result in the more signal transduction for cell proliferations and amplifications lead to the tumor formation. The mutated ROS protein have reduced GTPase activity. ROS mutation can occur in 3 different proto-oncogenes like KRAS, HRAS and NRAS leads to various carcinomas. Most commonly, KRAS mutation can lead to pancreas carcinomas. NRAS mutation can found in some hematologic malignancies. C. Activation by translocations. Translocation is the process of transfer of segments between the 2 chromosomes. The translocation process can do the activation of proto-oncogenes, but it shows less common. The best studied translocation in the formation of chromosome called the Philadelphia chromosome. Philadelphia chromosome will found in the chronic myeloid leukemia, that is called CML. The translocation occurs between 2 chromosomes, chromosome 9 and chromosome 22. Chromosome 9 consists of ABL oncogene at the position 9q34 region, where the break occurs during the translocations. Chromosome 22 consists of BCR oncogene, where the chromosome 9 will get fused with it. This will produce the chimeric protein, which includes high tyrosine kinase activity with the unregulated cell localization property. This translocation can lead to the acute lymphoblastic leukemia ALL. Activation by translocation can also lead to the formation of active region on the chromosome, which shows high transcription rate. The best examples studied is Burkitt's lymphoma. The translocation occur commonly between chromosome 8 and chromosome 14. Some other forms of the lymphoma can show the translocation between chromosome 2 and chromosome 8 or between chromosome 8 and chromosome 22. These involve the oncogene called MYC. In this case, there is no formation of chimeric protein. Instead, the activation of oncogene is found by close contact with the immunoglobulin locus on the other chromosome. In this case, the immunoglobulin locus IGH is present at the long arm of chromosome 14, 14q32. The immunoglobulin locus IgK is present at the short arm of chromosome 2, 2p12, and IgL is present at the short arm of chromosome 8, 8p24. Placing MYC near the IgH locus makes the activation of MYC, which encodes the protein involved in the regulation of cell proliferation. This translocation will lead to the uncontrolled proliferation of the cells in Burkitt's lymphoma. Oncogenes and the cancer. In many different types of tumors in human showed the presence of active oncogenes. It was found that many gene transfer experiments identified the presence of oncogenes. In 1981, scientist Weinberg identified the cellular oncogenes in human bladder carcinoma. These cellular oncogenes later found in many forms of the cancer in human. In the initial days from the gene transfer experiment, the oncogene RAS was identified for the first time in human cancer cells. In many forms of tumors, the oncogene found was cellular homologs of oncogenes, and in some tumors it was found that the new active oncogenes. RAS is the homolog of the RAS-H oncogene. A RAS-H oncogene is present in the sarcoma virus. There are many forms of RAS oncogene family found from the malignancy in human. These oncogenes will not present in the normal functioning of cells. These oncogenes mainly form by the mutation that occur in the tumor cells. A point mutation can change a single amino acid in the sequence by deletion or substitution, result in the formation of mutated proteins. By altering the signaling pathway, the binding of GTP molecules mainly regulates ROS gene functions. GTP binding makes ROS to remain in the active state. During this active state, these genes help in cell proliferation. Once the work is done, they get into the inactive state by the GDP binding. In the case of tumor cells, these oncogenes will not show the hydrolysis of GTP. The ROS proteins will remain in the active form by binding with GTP itself. Hence, there will be a continuous cell division occur without any stimulation from the growth factor. These will include activation of mitogen activated protein kinase called as MAP kinase pathway. As mentioned earlier, the many forms of cancer are caused not only by point mutation, but it can also be by chromosomal rearrangements like translocations and gene amplification. These can occur in different pathways of cell development processes, and many other oncogenes are involved. Roles of oncogene products. The gene products of the proto-oncogenes help in normal cell proliferation. The oncogene proteins show its role in the unregulated cell deaths and uncontrolled proliferation of the cells. They can also act as molecules like growth factors in the signaling pathway for cell proliferation and the survival of the cell. Some oncogene products can also act as the growth factor receptors like tyrosine kinase receptors on the cell membrane and receive the signal for cell proliferation. In the case of cancer, the tumor cells can produce essential signal molecules for cell growth. These molecules can act on the same tumor cells that are autocrine signaling and helps the tumor cells to grow more rapidly. Some of the proto-oncogenes will encode for the protein like cyclins. The growth factor stimulations can make the cyclins to progress the cell cycle. Some of the thyroid hormones and the retinoic acid show their role in cell differentiation. They will act inside the cell and bind to the intracellular receptors and regulate the internal transcription process. The mutated forms of these cells will serve as the oncogene, result in the unregulated cell differentiation. To conclude, nowadays different forms of malignance are establishing. Avoiding the potential cause of cancer is the simple method of prevention. Thank you.